It is clear.
Okay, thanks. Okay, good morning, everyone. We will have a quick review of what we have learned in the last class. We have been learning about mutations. We had a good discussion or review on the amount of what change means and how it can affect and what are the consequences. We had a brief introduction to it. And we have also started learning a few things. I'll have a quick review. And the change can be insertions, deletions, inversions, duplications, and so on. And in that, we also have substitutions. Substitutions can be two, that is transition and transversion. Purine to purine or pyrimidine to pyrimidine is transition. If it is vice versa, that is purine to pyrimidine or pyrimidine to purine, then it is transversion. And we are learning about replication errors. You can consider them spontaneous because it's not because of an external agent that causes these mutations. It is happening by normal or natural processes where DNA polymerase may have some error rate, which might escape proofreading, and then they become proofreading by DNA polymerase. But also there are repair mechanisms. Some of these mismatches can escape that mismatch and be established as a mutation. We have also defined what is meant by mutation. In terms of genetic information, mutation entails two things. The first one is that it has to have a genetic change in the sequence. And the second part is that it should be inherited. Inherited doesn't mean it has to be from one organism to another organism. It can be from one cell to the other cell, okay, to the daughter cell. That is mutation. So we also had a look at what is slippage. When there are repeats like this, either the template or the primer can slip, and it could result in insertion or deletion. So how does a replication error become a mutation? Say during replication, DNA polymerase has made a mistake. For example, as in this case, a G instead of T. But it is not a mutation yet, because here is something, the original one or the native sequence, that is A, whereas in this place it is a mutation. So it is something like half a mutation. If this particular mismatch has escaped the repair mechanisms, then and undergo replication, then it becomes a fully established mutation, as you can see here, CG, right? That is how a replication error becomes a mutation. But otherwise, normally what would happen is repair mechanisms will make it to the native state. I think we are supposed to have a discussion from here on. So along with replication, as long as the replication is going ahead, say for example, in this case, say if this is a DNA and replication fork, sorry, 1 second. Say this is replication fork and this is assumed DNA, newly synthesized one. And these are the proteins involved in replication, and they are moving in this direction. As soon as the replication is finished, there is a repair mechanism dedicated to correcting all the mismatches that are there, that are as a result of replication. So repair mechanisms will come by and will keep scanning this DNA, the newly synthesized DNA like this. It is something like following the replication fork, and then it will keep correcting if there are any errors, mismatches. Okay, and that is called as mismatch repair system. Okay, the first thing, objectives, they scan the genome for mismatches. How do they scan? I think we have a picture somewhere, but we will try to do that. So the objective is to find out where there are mismatches in the DNA, and the second one is to correct the mismatch. So it has multiple components. Those are referred to as by these proteins, MutS, MutL, MutH, and then you have UVRD, and so on, and DNA polymerase 3 and ligase. So we will see what each of these proteins do. MutS is a dimer, like is shown in here. MutS is a dimer, and it binds to DNA, something as shown in this picture here, like this. It encircles the DNA and keeps on scanning the DNA. It keeps moving in this direction. If it encounters a mismatch like this, here the mismatch is T and G, right? If you can see it, the mismatch is T and G. So that is a mismatch which is required to be repaired. And that is, anytime mismatch occurs, DNA is distorted a little bit. I hope you can see that if they don't form hydrogen bonds properly, then there is a distortion in the DNA. And at those locations, MutS binds, and there is a conformational change because of the distortion in the DNA. Where does conformational change in? MutS. Say, because I hope you see that the shape between this one and this one is different. And also it includes the binding of ATP. Right? So now the repair mechanism has scanned and located itself at the place where there is a mismatch. Then comes the second proteins, that is MutL and MutH. MutL binds onto the 3' side, like in here, or you can just say right now at the left side. And MutH is the one that creates a nick. What is a nick? It is a breakage of phosphodiester bond, right? And so you can see that where does it nick? MutH does not nick anywhere just like that. It recognizes the methylated and unmethylated GATC regions. Which one is the methylated one? The parent strand or the newly synthesized strand? The parent strand is where methylation of GATC sequence happens, and that is done by DAM methylase. I hope you remember when we have learned about it, about the regulation of replication in E. coli, right? GATC is there. We learned about what is meant by hemimethylation and so on. So here, the same DAM methylase, methylation of GATC regions also helps the strand, helps the DNA or the MutH to cleave one of the strands. You can see that, say, G and T are here. Which one should be removed? Should this G be removed or should the T be removed? Which one is the actual sequence that is supposed to be, what is in the parent strand is the true one? What is mismatch happened in the newly synthesized strand? So the replacement of the newly synthesized part should happen specifically. Say, for example, if MutH has cleaved here and removed this part, is the repair mechanism repairing the DNA or establishing a mutation? Right? That is how the repair mechanisms have evolved to bring about changes, to bring about or repair the mutation or mismatch that happened in the new one. Right? That is a beautiful thing. Otherwise, the evolution of these repair mechanisms would have been promoting mutations instead of removing the mutations. Okay, I hope you understood that. So MutH and it has made a nick, and there will be an exonuclease activity which will degrade this DNA from here to here, like this, right? And now DNA polymerase will take over this place. This is the 3' OH. That is where that is where DNA will be synthesized, and eventually A will be added in this location, which otherwise there was a G here, and now DNA ligase will seal the gap. So for the exonuclease activity, you need this strand here to be separated, and in that case, you need UVRD helicase activity. Okay, quick review. We will see mutase. The function of mutase is to identify the mismatches. Anywhere there are mismatches, there is a conformational change in mutase. There is a binding of ATP, and then uh, there is loading of mut-L and mut-H. Mut-L's function is to load mut-H. So mut-L makes the interaction with mutase, and then mut-H binds to that place and makes a nick. And then UVRD, using its helicase activity, will unwind the DNA here, and it is excluded by exonucleases. And there will be a second nick, and then eventually there will be filling up by DNA polymerase, and addition by using ligase, the DNA is sealed. Okay. Yes. So there are many other things, as I was discussing in the previous slide. If you didn't understand that, I hope you will try to understand here. Say this is a DNA, and okay, assume this is the parent DNA, and then we have two smaller new DNA molecules have formed. Assume that that gave rise to this. Okay. So the blue ones that I'm drawing here are the ones that are newly synthesized. Newly synthesized strands. The red ones are the parent strands. So if the question that I was telling you is there is A and there is G here. Sorry. Yes, say there is A and here is G. The parent DNA had A and T. Now there is a mutation, and I will not say there is no problem in the second DNA molecule. In the first DNA molecule, there is a mismatch. Okay, what is the mismatch? A is base pair, or A is in the location or complementary or in the opposite side of G. Which one is the wrong one that is added? Typically, the DNA, wherever GATCs are there, you would have had methylation like this, right? The green, whatever I'm drawing, is apparently assume it as to be the DNA, methylation. So if you look after replication, only the parent strand, the one that we had, will actually have the methylation, and the other strand will not be methylated. Sorry. Right, now I did correctly. Something like this. So if we have to remove and we have to replace something, replace one of the strands, and that is supposedly this is the G that is wrong, right? I hope you remember what is meant by hemimethylated DNA, right? Only one strand of it is methylated, whereas the newly synthesized or the other strand is not methylated. So any changes, any repair that should be done will be done to the newly synthesized DNA strands. So what will happen is mutase will come, recognize this location, and then you have mutel, mut-H, they will make a cleavage, there will be exonuclease activity, some bases, not just that one base, there are actually more number of bases are removed here and there, out on either side a little bit, and then you have the synthesis of new DNA from here, taking that 3' polymer, 3' OH, DNA polymerase will synthesize, and the ligase will seal the gap. Yes, that is about mismatch repair mechanism. This is the same thing illustrated again, and how mutage actually recognizes and so on. So you can keep always mutage can only make a nick on the unmethylated strand. That is what you should remember. So there is a problem then, right? The problem or a logic to understand. If this is mutage, mut S, and you have mutal, one second. Yes, mutal is recognizing the methylated DNA, and that is bringing in also along with it mutage, and then a nick is being made. How is the DNA from here to here cut? That's the question. So there is XO7 or RegJ, they call it, which is a 5' to 3' exonuclease activity. It will chop off the DNA until about here. Okay. What is the exonuclease activity here? It is 5' to 3' exonuclease activity. Then we have DNA polymerase will come and will then correct it, and then ligation will happen. That is fine. What if the mutation is on this side? What I am trying to imply is, as long as the methylation is here, methylation, then mutal is working. What if there is no methylation on this side, but it is present on this side? What if that is the scenario, what we are discussing in this case? Here, in this case, the mutation is here. This is the mismatch that is present downstream. Okay, I have to also tell you about what is meant by downstream and upstream. Say this is a point. All the things that you're talking here after a point, then you are talking about, you're saying as downstream. And if you're talking about, this is your reference point and you're saying it this way, then you're talking it as upstream. Okay, this is a terminology that we will use in the book, and that's the standard terminology used in molecular biology. So here, sorry. Yes, so as long as the methylation, or sorry, the mismatch is downstream of this methylation, then it is all right. You will have a nick made by mutage, and then XO7 exonuclease activity will use, that is 5' to 3' exonuclease activity, will chop it off and remove, and it is replaced with a new strand, new DNA. What if the mutation or the mismatch is upstream of the methylation? Then you need a different type of exonuclease activity, because the exonuclease activity required is, or degradation of the strand should be from 3' to 5', isn't it? So we have a different exonuclease that's called as XO1. It has 3' to 5' exonuclease activity. It will remove it, and then it is filled up by DNA polymerase and then sealed by ligase. Okay, I hope you understood the point of it. So eukaryotes also have a mismatch repair mechanism, and there are several proteins. I'm not going to discuss in detail. I will go to one point. They are called as MSH, MSH, MSL. These are the proteins, and PMS, and so on. So you are supposed to read the textbook. A question will come or may come, and you'll also have to read about some of the diseases that are implicated or because of repair mechanisms. If we don't have repair mechanisms, then the number of mutations we accumulate, or a cell accumulates, is very high, and then it might become cancerous. And therefore you should try to understand what kind of diseases will occur if there is a mutation in the repair mechanism, right? Deficiency of repair will cause to enhanced or enhanced aging and death, also cancer, and so many other complications. One thing I would want to focus, of course, is that how in E. coli we have, based on DAM methylase and the methylation of GATC sequence, we are able to recognize um, which one is the parent strand and which one is the newly synthesized strand, so that they are able to correct the newly synthesized strand. In eukaryotes, this dimethylase thing is not there. Okay, that kind of differentiation is not there. Instead, they would always prefer something like this. Uh, if we have a newly synthesized strand, say this is a parent strand, and newly synthesized strand are two types. One is the lagging strand, which is continuously synthesized. The other one is the lagging strand. One is the leading strand, one is the lagging strand. Lagging strand is discontinuously synthesized in the Okazaki fragments. So if there are any Okazaki fragments, that means that is the DNA that is newly synthesized. So any repair changes that are to be made will usually be made on the um, on this one and on the Okazaki fragments and sealed up. Then what about the leading strand? In that case, I hope you remember what is meant by PCNA. What is the function of PCNA? It is a sliding clamp. In eukaryote, it is referred to as PCNA is the sliding clamp. What is the function of sliding clamp? It is to increase the processivity of DNA polymerase. So sliding clamp typically will be bound around the DNA like this, and then it will and to which the DNA polymerase will bind and will keep polymerizing. What is the full form of PCNA? Proliferating cell nuclear antigen. Okay, so remember that. So the DNA PCNA will have an interaction with the parent strand, right? It's always with the parent strand because um, because that is where the DNA polymerase is. Say, for example, I will try to erase and redraw. Sorry. So we'll quickly, I'll just quickly try to draw it. So we have a DNA strand and assume that this is the 5' to 3', and the lower strand is being synthesized, and that would be, say, if I draw this will be 5' and 3'. So DNA polymerase will be moving, adding more and more DNA bases this way. That means the PCNA will be bound around that DNA here, the parent strand, and DNA polymerase is here, adding more and more bases as it progresses in this direction. So those that are in interaction with PCNA will also be able to distinguish which one is the parent strand, which one is not the parent strand or the newly synthesized strand. Okay. And also you remember this, please, that as we keep going into more and more details, we will reach levels. Say this is a textbook, and if we keep on going into depth, we will reach, this is the area of, say, research. That means we are still trying to learn, and this is where this is something like the unknown, right? If you take the textbook that we are discussing, it will try to encompass more and more that is in the current research. That means there are some things yet to be found, not yet clear. We have understood partially. So textbook is this, where it's supposed to be completely established, but of course it's relative. And this is the area where we know partially, that is where research is being done. And there are many more things in the future that we don't know, right? We will keep on pushing these boundaries, and that is what is a PhD or research. You don't know what it is and you're trying to find. In the textbook also, there will be several things that tries to incorporate the current research. We know some things, we don't know many things. So, and there will be some things that are bound to change also. So try to therefore my emphasis will always be on the principles rather than the exact facts. So the logic of how things happen is what you should try to know. Okay, crashed again. 1 second. I have to sort this out. This has some crashing issues. Yes, where are we? So that is about DNA replication, errors, and mismatch repair mechanisms. We have also learned about slippage, in which additions and insertions and deletions can also happen. Now we are trying to learn as what happens to DNA. In a cell, we have the DNA, and that DNA is always bombarded with, or there are several other chemicals within the DNA. That means the the implications or the interactions of the other molecules on the DNA is always possible, right? Because of which DNA is going to be affected. There will be changes, there will be damage to DNA. And that is what we are going to learn from now on for a while. These are kind of spontaneous because there is metabolism happening. Because of metabolism, some byproducts are formed, which may influence the DNA. Okay, so those are all, there are spontaneously some things will happen, that is hydrolysis. When you say the bases can, I think in the next slides we will check that out, where there are also some loss of whole base can happen, loss of some groups may happen, something like deamination, and that is also a damage, right? Demethylation, if it happened, that is also a damage. Say for example, what we learned about uracil. I think it's, as I keep mentioning it, 1, 2, 3, 4, 5, 6. And uracil is this, right? If you have a methyl group, then it is thymine. But assume that there is this methyl group demethylation happen. What is the group then? It is uracil, which is a damage, which is not a normal base within the DNA. So that's also DNA damage. There are also other damage that can happen because of chemicals, additional chemicals from outside, which could be alkylations, oxidation, radiation, and so on. And base analogs, those that look like bases. Analogs means they are almost similar to, in structurally, they are similar to that of some of the nitrogenous bases that are part of the DNA. They also can cause mutations. And there are intercalating agents. We will talk about it, what is meant by intercalating agent. Ethidium bromide that we use in electrophoresis of DNA. For visualization, we use ethidium bromide, which is also an intercalating agent. So the first thing is hydrolytic damage. It can happen, as I said, a base can be lost or a group can be lost. Spontaneous deamination. Say, for example, cytosine, as I was telling, yeah. Amino group is here, and this amino group is lost, and a O oxygen atom is added here. Now it formed because of deamination of cytosine results in uracil, which is a abnormal base within the DNA, right? And there are also where this glycosidic bond, I hope you remember that this is the base and you have the deoxyribose in this case, like this, right? And if this glycosidic bond is broken, then you are losing the whole base itself. That is depurination. Why are we saying purination? Because guanine is a purine. Similarly, we can also have a methylation. Sorry, here is 5-methyl cytosine. That is a nice one that we will discuss as we keep progressing. When it comes to epigenetics part and the last part of gene regulation in eukaryotes, we will learn about epigenetics. There we will know that some of the DNA, most of them are cytosins. are methylated. Okay, it has some regulatory functions right now, just remember that. Where is the methylation? It is on the 5 prime. So 1, 2, 3, 4, 5. Fifth carbon is here. I will try to focus a little bit more on this so that you will understand it better. So here is cytosine, 5-methyl cytosine. This is the methyl group, and cytosine has an amino group and O here, and you have a methyl group that is added by DNA methylases, methyl transferases. So if I will also try to show this part, what happened here? Deamination of this, of the amino group on cytosine is resulted in uracil. It is an abnormal base, you know it, we all know it, repair mechanisms know it, so they will remove the U and uracil and replace it with a correct base, that is cytosine. What if a methyl cytosine is deaminated? Then you end up forming thymine. And thymine is normal base of DNA. Then which one should we repair? What I'm trying to say here also is that I will try to put this way, or I will just draw a sequence. You try to C, G, G, C, G, A, C, and this is MC, that is methylated C. Okay, I'll just put it as this one, and G, A, T. Okay, that is, I hope it is clear. I'll make it a double strand for understanding. C, G, T, and here it is G, C, T, A. There are 2 cytosines here. This is the first one. Here is one cytosine, and the other one is methylated cytosine. Now we will do deamination of both the cytosines on the upper strand, this upper strand, and let's see what would happen. So we will get G, and deamination of cytosine will result in uracil, G, A, and deamination of methylated cytosine will result in T, right? So just we'll put it like this so that we don't get confused with that. G, A, T. What is on the lower strand? C, G, C, T, G, C, T, A. Focus on the differences between, this is one uracil, that is deaminated cytosine. This is a thymine, that is deaminated methyl cytosine. In these two, in this case, which one is the wrong base or damaged base? It's easy to figure it out because uracil is not a normal base. So what will repair mechanisms do? They will just remove this part, uracil, because that's abnormally located, and then they will add a cytosine here. Repair mechanisms have done a good job there. What about this case? Which of it is abnormal? Is T abnormal or is G abnormal? It's very difficult to find out, right? It's almost impossible because G is normal base, guanine, as well as T is also normal base. Then there will be a difficulty in which one to repair. That is when, if deamination of methylated cytosine happens, as I'm showing here, then there will be a lot of mutations that might happen because the repair mechanisms cannot distinguish between which one is the mutated base, because deamination of methylated cytosine results in formation of thymine, which is a natural base within the DNA. Okay, so that is about hydrolysis. And there are many other, the other chemical modifications that happen are alkylations. They can happen on the N or O, that is transfer of, alkylation is transfer of methyl groups or ethyl groups to some of the sites, something like nitrogen or oxygen like this. In some other places, oxidation may happen of any of these. Deamination may happen. Alkylation may happen on different bases. This is just showing one base. Addition of any of these things could result in bulkiness of DNA, or can interfere with any of the processes, or even sometimes base pairing also. Okay, so there will be then we will have difficulty with, and then should remove the bases. As I said, they interfere in even in base pairing. If we had an oxo G, I think it should be here. Okay, yes, we will just take it this way. Oxo G, that is oxidated on any of on this position here. Let me see, this picture is better. So when it base pairs with oxo G, oxo G can base pair with A or C. Okay, so what would happen in that scenario is, if you had a G, A, C, G, T, A, assume that this is the template 5 prime to 3 prime, and assume also that this one, the guanine here, is oxidated. Then if the DNA polymerase was synthesizing, this will be the 5 prime for the lower strand, T, A, and it would be base pairing with A or C. If it were base pairing with C, no issues. G normally base pairs with C, that is all right. But sometimes it may also be, it is something like tautomers. It can base pair with a different base, that is A. It can also base pair with A. Of course, this is a purine and also a purine, and they are going to be bulky. And assume that this is going to get continued G, T, C. This is 3 prime. Now you have oxo G and A. Which one do you think is the abnormal one? A is not abnormal. If repair mechanisms have to come and check out which one is of these two is bad or wrong, A is fine because it's a normal base. What about oxo G? That seems to be something damaged. And then if the repair mechanisms happen to remove this G, and what would they add here? They would have added T here. So what was GC base pair has become a TA base pair. That is a mutation, isn't it? So like that, many changes, any of these changes can influence the DNA, and they keep changing. As I was telling you again, change is constant, and there is nothing that is going to be static. Cultures, languages, religions, many things keep changing, and that is what happens to DNA also. Material or immaterial, everything changes. Here is a test developed. It is called as EMIS test. It is about to test the ability of a compound. Now we are going to discuss about induced mutations. What were the things that we were discussing till now? They are spontaneous mutations. Spontaneous mutations include replication errors. They also include deamination or depurination and effects like that. We were supposed to discuss about base analogs, intercalating agents, and so on, or radiation and so on, that induces from the outside induces change in the DNA. Those are all called as induced mutations. We need a test to find out what is meant by how to see the ability of a compound to induce mutations, right? The degree of mutagenesis. So what is a mutagen? We will have some vocabulary. Mutagen is a compound or a physical agent. It can be UV radiation, X-rays, and so on also, and chemical compounds as well. Mutagen is the one that can cause mutations. Okay? And mutagenicity is the ability to cause mutagenicity. The degree of mutagenicity, which would mean the number of mutations it could cause per amount of matter, something like that. What is carcinogenicity? Carcinogenicity is ability to cause cancer. Mutagenicity is the ability to cause mutation. What is the relationship between mutation and cancer? All cancers have mutations, are caused by mutations. There are subtle change differences as we progress into real causes of cancer, but almost all the cancers we know of have mutations in some form of mutations. But there can also be mutations which actually do not cause cancers. So you can say something like this: all carcinogens are the agents that can cause cancer. All carcinogens are mutagens, but all mutagens are not carcinogens. Say, for example, you had a cell, and within the DNA of it, there is a mutation, but it is not cancerous, right? A mutagenicity and carcinogenicity are slightly different in that sense. And here are some classic microbiology terminology that you should remember. What is an auxotrop, prototrop, minimal medium, and then we will know what is, we already have discussed about spontaneous mutation, and we will discuss about spontaneous revertants in a while. For this, to understand Amy's test, we should have, this is based on bacteria, right? That is, and there is terminology used again. What is an auxotrop? Auxotrop is something like the, we will first talk about what is a prototrop. Prototrop is the wild type. Okay. What do you mean by wild type? When you say wild type, it means that can grow in the wild or in the nature. Grow in nature. If it can grow in nature, it should be able to use the materials that are available in the nature by itself and synthesize everything it requires by itself, right? That is called a wild type. Then what is an auxotrop? Auxotrop here is a mutant. That means it has lost one or more genes and therefore cannot synthesize something. So you put this in nature, it will die because it has lost the, say for example, lost the ability to synthesize some amino acid, or lost the ability to metabolize lactose, for example. Those are called as auxotrops. That means there is a deficiency in metabolic genes. Deficiency in metabolic genes. Metabolic genes. Whereas wild type has all the genes. Okay. So I hope you understood what is a prototrop and what is an auxotrop. Prototrop is the wild type. It has all the genes required to grow in the nature. Whereas auxotrop is a mutant. It may have deficiency in one or more of the metabolic genes, so it cannot grow in the nature. But if we supply, say for example, this mutant auxotrop has deficiency in X, in producing X, it cannot make an amino acid. So if we provide that X in the medium, then it can take up that molecule and then grow well. In the lab, we can culture auxotrops. Okay, now let's also see one more thing that is called as minimal media. There are two different types of media. There are several categories of media, but for Our purpose, we will talk about what is meant by minimal medium, and rich medium, or it's also called as complex medium and so on. Minimal medium is something, say in rich medium, we provide proteins. Medium here is for if we want to grow bacteria in the lab, we will make petri plates. We'll take petri plates. We'll put in nutrient agar. Agar is for solidification, or it will solidify and stay there. And along with that, we add some nutrients for bacteria to grow. Bacteria take up those nutrients and then grow on the surface, something like this. It will form colonies. One bacterium will form one colony. But if you over populate it, then they will grow into complete mat of cells, right? But for their growth, they need nutrition. How we provide the nutrition is what may differentiate between minimal medium and rich medium. I think based on your biochemistry knowledge, you know that all of the molecules would be made of C, H, N, O, P, S. Carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur. There will be many other elements, but these are the majority of the elements are, they belong to these, right? If I'm giving a protein, then I'm giving CHNOS. If I'm giving nucleic acids, then I'm giving almost all except the sulfur. I think we discussed this in Hershey-Chase experiment, the blender experiment. So how we provide for if one bacterium has to become two bacteria, then that means it should have had the supply of all these elements along with many others. So how we provide them? If I'm just giving it as simple compounds means water, ammonium chloride, like that. We are not giving compounds. On the other hand, look here what I'm giving. Proteins. You can give nucleic acids. Just some extract you take and you just dump it in there. You don't have how much, you are just giving it in kind of organic form, right? Whereas in the minimal medium, you're giving in inorganic form. Only compound that is common in both of them would be glucose, GLC. GLC is the short form for glucose. Don't ever write it as GLU. GLU is for glutamic acid. That's an amino acid. For glucose, you will write as GLC. Okay. So I hope you understood minimal medium and rich medium. Rich medium, we are giving compounds in organic form so that they can easily take up. You're giving it as amino acids and so on. Amino acids, proteins, nitrogenous bases, and so on. You can give, bacteria will take it happily and grow. But if you are giving all inorganic forms like this, then the bacterium should take up all these, synthesize the compounds, that is amino acids, proteins, everything, and then only it can form, divide and form two daughter cells, right? So if we talk again about auxotropes and prototropes, let me erase and show what will happen. Auxotropes can grow, prototropes can grow in rich medium, right? Because you're giving all in good form. It's like early morning, you give a good breakfast, and it's readily going to take it up, eat it. But if you give minimal medium, because prototrope has the capacity, something like you give the ingredients to make a breakfast. It has to make all the it has to prepare everything and then put on the dosa or idli, whatever, and then make it and then consume it. Prototropes have the energy, capacity, and everything to do that, genes mostly. So they can grow on minimal medium as well. What about auxotrophs? You serve the breakfast, they're happy, they will eat. Analogy, take it that way. And but if you give minimal media, then they don't have one or more of the capacities to make what it requires to grow and divide into two cells. So they fail to grow in this. Okay, so they cannot make the food ready for itself. You have to provide ready-made food for it, auxotrophs, because they have deficiency in one or more of the genes. Okay, so try to remember this, and I will give a brief introduction to this so that when we discuss tomorrow, you will be able to understand better. Don't worry if you don't understand what I'm going to say now. It's all right. Salmonella, they take a bacterial culture and Salmonella. They took an auxotroph of Salmonella. What is it auxotropic for? It cannot synthesize histidine. When you say like this, his minus, that means this Salmonella cannot synthesize histidine. So will it grow on rich medium? Yes, because histidine is already prepared readily, so it just takes up and uses up. Can it grow on minimal medium? No, because there is no way it can synthesize histidine. So it cannot grow on minimal medium. Okay, so here is Salmonella, which is his minus, and you take about 10 to the power 8 cells, and you have added into culture plates like this, Petri plates. But one thing is, they did not add any histidine. What will happen then? They shouldn't have grown, right? So you have, there are two plates, plate 1 and plate 2. In plate 1, you didn't add anything. I hope you realize that EMIS test is for to estimate the mutagenicity of a mutation. Okay, so in the other plate you add mutation and see how many mutants have formed. These are spontaneous revertants, whereas there are more number. If this compound that is added is not a mutagen, you will find maybe 2 or 3. But if you're finding something like 10 or 12, that means the compound that you added in this plate must have made those mutations. That is why that these revertants were happening. That's all right. I'll stop here, and I will take up from here for next class EMIS test, and we will discuss a few repair mechanisms in the next class. Okay. If you have any questions, please do post